I'm Simone. Thanks, Paul, for the introduction. So thank you to Cure Cancer for holding this event today and for funding this project. I'm just going to start by giving a brief overview of who I am and where I come from. Sorry. Okay. So I work at the Doherty Institute in Melbourne, and last year I completed my PhD at the Doherty Institute at the University of Melbourne. And during that time, I was working on a particular type of immune cell, which is called a resident T cell, and how that cell can protect against cancer. And so this year, I started as a postdoc also at the University of Melbourne with Laura McKay. And this is where Cure Cancer sort of come into the picture for me, funding a project on how we can possibly target these resident T cells that I studied in my PhD for improving cancer immunotherapy. And then hopefully next, I'm going to be moving to the States, COVID permitting eventually, to continue working on cancer immunotherapy at the University of Pennsylvania from next year. So what is immunotherapy? So as we just heard, immunotherapy is a highly successful treatment for many different types of cancers. And there are many different types of immunotherapies. And one immunotherapy is CAR T cell immunotherapy, which basically involves engineering immune cells called T cells so that they can specifically recognize cancer cells. And upon encountering those cancer cells, they're able to target and kill those cells. So this CAR T cell therapy is one of these promising types of immunotherapy, and it's really excellent at fighting against blood cancers, but it's far less effective against solid cancers. And the reasons for this are basically, or sort of summed down to the fact that these cells often persist very poorly within solid cancers, within the tumor microenvironment, and they also have a reduced functionality in a lot of cases. And so we need strategies to improve immunotherapy against solid cancers, including CAR T cell therapies. And so the immune cell type that I work on, which is called a resident T cell, is kind of a special type of immune cell because unlike most of the immune cells that you'd be familiar with that are constantly migrating through the body in the blood, these immune cells are not circulating. They're found within tissues where these solid cancers develop and grow. And so an important feature of these immune cells is that they're very long-lived and they persist in the same spot for many years, and they maintain high function for these many years. And so importantly, these cells, we've also shown, and other people have now shown too, that these cells are protective against many different kinds of solid cancers. So they have a broad ability to protect against several different types of cancers. It's not just limited to one type of cancer. We've shown that these cells are really important to protect against melanoma, but other people have shown that these cells are correlated with improved survival in breast cancer, lung cancer, colon cancer, and many other kinds of cancer as well. So we believe that targeting these resident T cells could help to improve immunotherapy against solid cancers. And so in this project, we're particularly focusing on CAR T cell immunotherapy. And what we have been able to show so far is that CAR T cells are capable of becoming resident or forming resident CAR T cells. And so what we want to investigate in this project is whether we can increase the formation of these CAR T cells and how we might be able to do this. And so to do that, we're using a variety of gene editing techniques so that we can try and force this process of conversion from a CAR T cell to become resident or a resident CAR T cell, and thereby increase the number of these CAR T cells within the solid tumor microenvironment. And we're also investigating how we might be able to manipulate the growth conditions under which these CAR T cells are cultivated so that once they're infused into the body, they'll be more likely to form this resident type of CAR T cell. We'll get many more of these resident CAR T cells within tumors. And then as a consequence of that, we'll then ask if that increase in the number of CAR T cells translates into an increase in cancer protection against a variety of different solid cancers. Using some solid cancer mouse models that we have available to us in the lab. And so I'll just finish by thanking everyone that's involved in this project. It's just definitely still in its early stages. It has been a little bit slowed down by COVID this year, but hopefully we can start getting a bit more of it done now that we've returned to the lab. So I'll thank everybody that's involved, and also obviously thank you very much to Cure Cancer and the funding partners for funding this work. Thanks, and I'll take any questions at the end.